So today we are going to start transport mechanism. Now what are transport mechanisms in biological system and why they are required? Why are they required? And what are exactly transport mechanisms? Why are they required? What purpose do they serve? And what are the types of transport mechanisms we have learned over these years? The best and the most simplest example, transport of O2 and transport of CO2. Transport of O2 through body to body tissues and collection of CO2 and exhalate it out to remove it from the body. This is one example, okay? This is the most basic and simplest example. What else can you give? What are other examples involved in these biological processes? Transport, like? Yes. Removal of waste material. Assimilation of food. Transport of hormones, yes. The energy that we derive from food. What does intestines do? They absorb that thing. Okay? So that's another good example. That's a very, very basic example. But since it's just 1 or 2 days here at the college, so we'll be very lenient, okay, right now. We'll give only very basic examples. But as the year ends, please try to avoid these because at medical colleges level, these are very lame examples. Very lame examples. So, in what categories do we divide transport mechanisms? How are they differentiated? Now we have a basic idea that what are transport mechanisms. Now how are these divided? Transport mechanisms. Anyone? Please be seated and answer. Yes, you are absolutely right. Active transport and passive transport. Now, what makes a process active and what makes a process passive? Yes. It does not require energy. Yes. Active transport and passive transport. One requires energy and another does not require energy. What do you think which would be more efficient? Active transport or passive transport? Why? Yes, because energy is being utilized there. अगर आम भाषा में बात करें तो एक चीज आपको फ्री में मिल रही है और एक चीज आप पैसे दे के ले रहे हो. नेचुरल है, जिसके पैसे दिए हैं उसकी क्वालिटी बेटर होगी. दैट वुड बी मोर एफिशिएंट. ओके, नाउ कमिंग बैक टू द टॉपिक, कैन यू गिव मी सम एग्जांपल्स ऑफ एक्टिव एंड पैसिव ट्रांसपोर्ट?
एक्टिव ट्रांसपोर्ट सोडियम पोटेशियम पंप.
Sodium potassium pump, yes. Active transport, yes.
Passive transport, diffusion.
Yes, diffusion of gases is a passive transport. Any more?
Channel proteins like water, aquaporin.
Aquaporin is active or passive? Yes, it is passive, but it has one more classification. Can you give that? Yes, facilitated. Now the term arises. What is the difference between passive and facilitated? What is the difference between them? Yes. So I think this topic must have been completed in physiology. Huh? So you have started reading the books, huh? Okay, good. So this class has a good IQ. That surprised me. sorry. Now, which is you require more, facilitated or simple or passive diffusion? Can you give me some examples? How it is operating in our body? Where it is operating in our body? Facilitated and simple diffusion. Just I am giving you a brief overview. Yes, ma'am, please don't look outside. Class is not doing there. Can you give me few examples of that? The most simple for simple diffusion, the best example.
Exhalation.
Yes, she is absolutely right. Exchange of gases at tissue level also and in lungs also. It is driven by diffusion, simple diffusion. And for facilitated, few of you have given examples. Like, would someone like to repeat them? What are the examples of facilitated? At the back?
Solute transport ions across the
No. I am asking about the examples of facilitated. No, that is active.
aquaporin.
Yes, aquaporin. Sodium channel. Ion channels. Okay, ion channels. There is a difference between sodium channel and sodium pump. Can anyone tell what would be the difference between a sodium channel and a sodium pump?
In sodium channel, 3 potassium ions are thrown outside and 2 sodium ions are taken inside. Something like that.
It is in sodium?
Three sodium ions are
No, you are giving the example for sodium channel. No, that is sodium pump.
pumps.
Well, look at the term pump. Pump क्या require करता? Pump requires energy. It gives out 3 sodium and intakes 2 potassium against concentration gradient. That is why it requires energy. Because sodium concentration outside the cell is high as compared to inside the cell. Why? कहने को हम कह देते हैं it is a fact, but there is a very good reason behind it. Why? Come on, you can try. We are all here to learn only. There are no schooling punishments in this classroom. You can freely give any thought that is crossing your mind.
Sodium channel से अगर ज़्यादा सोडियम आयन से अगर ज़्यादा होंगे तो हाइपर
क्या हो जाएगा?
हाइपरटोनिक ऑस्मोसिस.
No. Look. Let's say this is a cell. Okay? Are you able to see now, बच्चे? At the back? Now, as per you, if we are throwing out 3 sodium out and we are taking 2 potassium in, there is a deficit of 1 ion here. यहाँ पे ऑलरेडी एक आयन का डेफिसिट हो गया है. 1 positive ion. There is a charge deficit also. Osmosis नहीं. Its reason is because life originated in sea. It is based on our evolution. Life originated in the sea. और समंदर के पानी में क्या होता है? नमक. क्या होता है? हाई कंसेंट्रेशन ऑफ सोडियम. So that thing is still being carried on to this day. Okay? इसका आंसर बहुत कम मिलेगा आपको. Thanks to my professor that he taught me this. Now let's come to active transport. I'm just giving you a brief overview. One by one I'll explain each and everything to you. Okay? Examples of active transport. One is just I have demonstrated. Sodium pump. Okay? Sodium pump. Channel facilitated होता है. Channel is facilitated. It does not require any energy. One example is sodium potassium pump. What are the other? Can you give some examples of active transport? Naturally, if there is a sodium pump, there will be a calcium pump. कहाँ होता है? Yes, muscles. Well, this class is impressing me. This class is good. Quite impressive, students. In muscles. And what is the function in muscles? Yes. And since we are sitting in a medical college, what would be its clinical application? Yes. You are in the right direction, but not there. Think. Now, since there are calcium channels also, which requires ATP for transport of calcium. So, can you tell me one clinical application? इसका medicine में कहाँ use हो सकता है? How can we exploit this in medicine?
किसी को cramps आ गया तो it must be because of deficit of calcium.
No, you all are thinking in the right direction, but you are not exactly there. No. Heart attack. Treating myocardial infarctions and heart attack. Treating hypertension. Okay. There are certain drugs which selectively blocks certain channels. I should not say channel, I should say pumps. Even there are drugs for channels also, but I'll just keep a very, very slow pace at your first lecture, okay? Now, when the pumps are blocked, what will happen? Certain amount of certain ions will not be permitted across the membrane, okay? And that will produce the desired effect. कोई हाइपरनेट्रीमिया का पेशेंट है, सोडियम एलिवेटेड है, so naturally we have to act on that sodium pump. Okay. Now there is one more thing, diabetes insipidus. Diabetes insipidus, है ना? What is diabetes insipidus? What is diabetes insipidus? Yes? Yes, excess loss of water through urine. But why it happens? What is the pathology behind it? Vasopressin. Vasopressin. So you are all aware with diabetes insipidus, है ना? It is also due to a channel defect, aquaporin. When vasopressin is in short supply, The aquaporins are not fully transcribed. So there is extensive loss of water through DCT. नेफ्रॉन के भी पार्ट्स होते हैं. DCT. DCT से रीअब्सॉर्ब नहीं होगा water. So it will lead to dehydration through continuous loss of water. What are other, you can say, applications of these pumps which are you actively using energy? Active pumps. Vision. It is one more classic example. You must have heard of that process, na? Depolarization in nerve conduction, repolarization, and then there is hyperpolarization. So image forms on our image forms on, actually you can say that image forms on retina, but actually बात ये होती है that retina captures the signal. Image forms here in brain at the visual cortex. ठीक है? So that process also require hyperpolarization, and hyperpolarization is achieved by pumps. Active transport is required there. So it is one more application. So now let's start. This was just a brief overview of the lecture, okay? First one is passive transport. First one is passive transport. What are the examples? Passive transport examples? Yes, osmosis is an example, but simple diffusion, osmosis. Now, what is the difference between simple diffusion and osmosis? No. ये तो पता होना चाहिए. You have just yes. Osmosis requires a semi-permeable membrane. देखो, osmosis is also a diffusion. What is the diffusing particle there? H2O, पानी ही तो diffuse कर रहा है. But the character which differentiate osmosis and diffusion is semi-permeable membrane. If there is a semi-permeable membrane, it is termed as osmosis. What is the next example after simple diffusion in passive transport? One is simple diffusion. Another is, she said, osmosis. What would be the next important one is? क्या? Facilitated transport. Number one is first part is passive transport. It is further classified into number one, simple diffusion. Which one? Number one is simple diffusion. Marker change कर दिया.
Yeah.
The second one would be, you can include osmosis in this only. Okay. You can include osmosis in this, or if you want to maintain a separate category, it's your choice. Both are correct. Second one is facilitated transport. Now this is very important. Now what is the difference between the two? Simple diffusion, facilitated transport. Now let's open up in the lecture. दोनों में फर्क क्या होता है? Since both are passive, दोनों में एनर्जी नहीं कंज्यूम हो रही है. We all know. Okay? But what is the key difference between the two? Yes. Facilitated transport requires a carrier protein. Example, aquaporin. Okay? मार्कर लाके दो थोड़ा जल्दी, क्योंकि बिना फिगर के एक्सप्लेन नहीं हो पाए. The best example of diffusion is exchange of gases. Okay? And the best example of facilitated diffusion is aquaporin, channel proteins. Now what are channel proteins? We have said that facilitated transport requires channel proteins. What are channel proteins? If this is a cell, na, you can wait a few minutes, they're just gonna change it. We require channel proteins for what? For transport. Okay? Because specific things cannot just straight away come into the cell due to presence of cell membrane or the lipid bilayer. सोना नहीं है? नहीं, आपके पीछे वाले को कह रहा हूं मैं. आप भी सो रहे हो? Presence of lipid bilayer. Due to presence of lipid bilayer, it acts as a barrier to most of the things which are present in the external environment. They cannot straight away enter it in the cell. And neither the things present in the cell, they also cannot go straight away into the external environment. That is why facilitated transport require channel proteins. Have you heard about the GLUT receptors? Yes? What are the GLUT receptors? Yes, what are GLUT receptors? Yes, yes, glucose transport, yes. Thank you. What are GLUT receptors? They transport glucose, but do they require energy or not? Quite impressive. Yes, they require insulin. अच्छा, एक और होता है जो इंसुलिन के बिना भी काम करता, वो कहाँ होता है? लिवर. लिवर पेरीफेरी में जो होता है, मसल्स होते हैं जहां पे ग्लूकोज का कंजम्पशन होता है सबसे ज्यादा, दे आर ऑपरेटेड अंडर द इन्फ्लुएंस ऑफ इंसुलिन. बट वेयर द इंसुलिन इज नॉट एक्टिव, दे आर प्रेजेंट इन लिवर. ऑन लिवर ग्लूट रिसेप्टर्स, इंसुलिन इज नॉट एक्टिव. Now let us suppose we want to transport these molecules inside. These are a kind of carrier proteins, okay? A kind of carrier proteins. What will happen that these would just attach here and would be thrown inside the cell. Simple. Simple. That's the process. But there is a catch in it. if you'll plot a graph between the rate of facilitated diffusion, there is one very interesting thing. Now, why this graph plateaus out? Flat क्यों हो जाता है ये आगे जाके? Saturation of what? Saturation of, yes, carrier proteins, because carrier proteins are limited. If there are 100 carrier proteins and you have infinite number of molecules which are to be transported, initially what will happen? 5 will get engaged, 10, 20, 50. Eventually all carrier molecules will start acting on their full efficiency. After that, no more increase in the rate would be seen. The rate of transport would become constant with time. Now, how can we use this in medicine? Since we are here in a medical college, do you find any practical application in medicine? Think. You do not require any heavy names. You just require a bit of common sense. Just look at that figure and look at this.
Sir, detect how much is the patient having enough carriers?
No. Carrier number does not influence that. Suppose I have a structurally similar compound or substance to be transported. Will it not compete with these active sites? It will compete, na? There will be competition as to who will acquire these sites, eventually affecting the rate of transport of these inside the cell or outside the cell, whatever the direction is. So this is called competitive inhibition. Okay? You will learn in biochemistry and pharmacology also in very detail about competitive inhibition. One example is succinate and malonate. That's a very simple example being given, okay? So far, we have covered simple diffusion, facilitated transport. Now what is left? क्या बचा तीसरा? Active transport. What is the classical feature of active transport? Against the concentration gradient. आप नल खोलते हैं तो आपके टैप में पानी आ जाता है, उसमें एनर्जी रिक्वायर नहीं लगती इतनी, लेकिन पानी ऊपर टंकी में चढ़ाने के लिए पंप लगता है, because that is against the concentration gradient. So what are the few examples which we have discussed in the intro part? Sodium potassium pump, or we can briefly say sodium pump, calcium pump, or vision. These are practical application. Vision. I'm just giving you very brief ideas of that. Sodium potassium pump. 3 sodium outside, 2 potassium inside against the concentration gradient. Okay, always remember कि sodium concentration cell के बाहर ज़्यादा होती है as compared to inside the cell. Okay, किसी ने ये नहीं पूछा how is the electrical neutrality would be maintained inside the cell? कैसे? How would be the osmolarity, osmotic gradient will be maintained? किसी ने बोला था यहाँ से. How? कैसे? सोचा है? हमारी सेल के अंदर, inside the cell, there are not only just ions, there are organic acids also. There are organic acids like बहुत सारे होते हैं, बहुत सारे, countless intermediates, countless. These are generated and destroyed as per the requirement to maintain the electrical neutrality inside the cell. Okay, how? Let us say this is any organic acid. Currently it is in unionized form. Let us say now there is a charge imbalance. Okay. Similarly, many more reactions could be performed inside the cell to maintain homeostasis. I hope you are familiar with the term homeostasis. What is homeostasis? To maintain steady state. तो एग्जैक्ट नहीं पता आपको भी हाँ, फॉर लाइफ प्रोसेसेस पढ़ो, थोड़ा सा पढ़ो होम्योस्टेसिस, ऐसे नहीं, थोड़ा कॉम्प्लेक्स है, इतनी आसानी से नहीं आएगा समझ में कि कुछ भी बोल दिया। अरे नहीं, मैंने क्या कहा, पढ़ो इसे थोड़ा सा। It has a small but very important and complex definition in homeostasis. So today's homework is look up what is homeostasis, the definition. पता हम सबको है definition, आप में से शायद ही कोई बताता है। Very complex, थोड़ा है इसमें, कुछ terms होता है। So this is one example of active transport. Now there is one clinical application of active transport, but before discussing that I want to ask one thing. Active transport is also divided into two parts. Do you know what are the two active types of active transport? No? Write down. First one is primary active. First one is primary active and second one is secondary active. Now what is the difference between the two? What is the difference between the two? Primary active and secondary active.
Primary is essential.
No. Secondary is also very essential. In fact, arguably more essential. क्या नाम है आपका? त्रिपुरा से, यहीं से त्रिपुरा से, कहाँ से? वेरी गुड, वेरी गुड. Energy is directly consumed in primary active, but it is not directly consumed in secondary active. I'll tell you in a minute. अभी इतना concept बिठाओ. तो अगर primary active में directly energy use हो रही है तो क्या है ये? It is a kind of primary active transport. Okay? Since it is directly requiring ATP, sodium potassium pump. Now, what is the example of secondary active transport? This is very important. Can you give the example of secondary active transport? One example. नहीं पता? This is a very classic example, and this is in fact a very good clinical you can say application also. You must have heard of one thing called ORS. क्या होता है ORS? ओरल हाँ, कब दिया जाता और किन्हें दिया जाता? वाटर लॉस होता है तो, और वाटर लॉस होता है, और देखो इधर देखो, I hope it is visible at the back, है ना? One sodium ion and one molecule of glucose. This has two sides. Okay, this channel has two sides. अभी हम क्या पढ़ रहे हैं? सेकेंडरी एक्टिव ट्रांसपोर्ट पढ़ रहे हैं. प्राइमरी एक्टिव नहीं, सेकेंडरी एक्टिव है ये. रिक्वायर्स एनर्जी. जब तक ये दोनों रिसेप्टर पोजीशंस ऑक्यूपाइड नहीं होंगी, चैनल काम नहीं करेगा. ठीक है, चैनल काम नहीं करेगा. द चैनल वोंट वर्क जब तक ये दोनों ऑक्यूपाई नहीं होंगे. रिजल्ट, सपोज हमें ग्लूकोज अगर चाहिए ना, तो साथ साथ में सोडियम भी फ्री में अंदर आ रहा सेल में. बेशक हमें उसकी रिक्वायरमेंट उस टाइम हो या ना हो, आइदर इट इज रिक्वायर्ड और नॉट, बट इट इज कमिंग इनसाइड द सेल ना, आ रहा है ना सेल के अंदर. सो इट चेंजेस द ऑस्मोटिक बैलेंस इनसाइड द सेल. इट चेंजेस द चार्ज न्यूट्रलिटी इनसाइड द सेल. नाउ दिस वुड हैव टू बी थ्रोन आउट. Well, you cannot throw the glucose molecule outside the cell. It's common sense. We require energy. Every cell requires energy. But sodium can be thrown out, है ना? Through sodium किससे बाहर जाएगा? Yes, sodium potassium pump. So this would be thrown out by sodium potassium pump or sodium pump, and that requires energy. तो एटीपी आपकी यहां लग रही है, यहां पे कोई एटीपी नहीं लगी. देयर इज नो एटीपी इन्वॉल्व्ड एट दिस प्रोसेस. A molecule of sodium and glucose can combine on a channel and can freely enter inside the cell. But at the moment we do not require sodium inside the cell. There must be some other better uses elsewhere. So sodium will be thrown out of the cell, and that requires energy, and that is called secondary active transport. क्या कहा जाता है इसे? Secondary active transport, because the energy or the ATP is utilized in the secondary step. Okay, that is the secondary active transport. Now open that portion of facilitated transport. Open it if you have written there. Just take a brief or short explanation on ion channels. On ion channels. Due to that marker issue, na, sometimes that chain of thought get broken. It's very easy to be seated there. Once you reach here, na, try, teach your fellow students. I hope you all are staying in the hostels, na. Do one exercise. Take 15 minutes. 15, not 50, 15. I know hostel time is very precious for each and every one of you, especially in the first year. Try to teach your roommates. If you are staying in 2 or in the 3, just make him sit there. Whatever topic, anat, physio, biochem, community, बैठ और मेरी बकवास सुन 10 मिनट अभी, और फिर जो भी गलती है बता क्या गलती है. This will enhance your skills in memorizing the topics. You cannot explain anything to anyone until and unless you yourself do not know it. And add the valuable add-ons from your friends' input. कि भाई यहाँ नहीं, यहाँ गलती है, यहाँ नहीं, यहाँ गलती है. Just take 15 minutes. A very good exercise. Now, ion channels. They can be of three types. Number one is voltage gated. Number two would be, किसी को पता है? What are the other types of channels? Ligand gated. What is a ligand? What is a ligand? Ligand क्या होता है? हाँ, क्या होता है ligand? बताओ. No. वो तो उसके बनने से coordinate compound बनता है. I'm asking what is a ligand, not a coordinate compound. What is a ligand? It is an electron donor. No. It is an electron pair donor. अरे मुझे 13 साल हो गए छोड़े हुए, अभी भी याद है. ऐसा ही होता है ना टेक्स्ट बुक्स में, कुछ है ना. अब ये प्लीज डोंट आर्ग्यू इट इज 2 और 3. देयर इज अ वेरी वैलिड रीजन फॉर राइटिंग इट 2. This would be donated. So this is a ligand, okay? Now, coming back to the topic, there are two types of ion channels. Voltage gated and ligand gated. There is one more category, but I am leaving it right now because it's your just first class today. I don't want to go all in the first lecture only कि यार biochemistry में तो कुछ मजा नहीं आता, बस ऐसे ही चलता रहता है. Okay? What are the examples of voltage gated channels? See, they are like that. If you want to discuss anything, I am here. You can talk to me. This channel is currently in the closed position. This is a voltage gated channel. See, there is a clear potential difference visible here. Okay, so this is in a closed state. Now to open this channel, what we'll do? We'll disrupt this. We will disrupt this. Disrupt this what? The potential difference. Since this is a voltage gated channel, what is voltage actually? It is a potential difference. It is a potential difference. अभी मैं यहाँ पे पैर रगड़ूंगा तो मेरे में कुछ चार्ज आ जाएगा. And this floor is at earth. Okay? तो कितना potential difference होगा? कुछ minus में होगा because I have gained electrons. Okay? So this kind of is potential difference. Once you disrupt this, this channel opens. Okay? The best example would be, what would be the best example? कहाँ देखते हैं? सबसे बेस्ट हिंट दिया. Origin and conduction of nerve impulse. Origin and conduction of nerve impulse. And this is a kind of channel coming under facilitated diffusion. Okay? So I felt this topic shouldn't be left. It's my duty to teach you. What would be the second example? If anyone want to try. पक्का? There are specific other ion channels also, which are specific for that. Specific ion channels are there. You will get them in physiology also, in biochemistry also. Okay. So let's leave it there. Now, second one would be the ligand gated channels. What is the second category? Ligand gated channel. These are a bit different. What was the trigger mechanism in voltage gated channel? Change in potential difference, not potential difference. Potential difference was already there. The change in potential difference. What was the trigger in voltage gated channels? Change in potential difference. Now, ligand gated channels are a different thing. Let us say it's a ligand gated channel, okay? Currently it is in closed position, okay? Currently it is in closed position. Now, the ligand gets attached in this region which wasn't occupied and the channel opens. Okay, this is how ligand gated channel works. The trigger mechanism is attachment of specific ligands. What are the examples? The best would be acetylcholine receptors, ACH receptors. So ACH acts as ligand. Okay. कान में जो मैकेनिज्म होता है हियरिंग का, उसमें कौन सा वो यूज होता है? सोचो. ना, we have generated the hearing impulse, but how the signal is getting transmitted? Through which? पक्का? You have any book with you right now? In hostel or anything? No. So your second homework would be to find out the channel involved in hearing mechanisms. What was the first one? To find the correct definition of homeostasis. Homeostasis. There is one more term related to that, hemostasis. सुना है hemostasis? क्या होता है? क्या होता है hemostasis? What is hemostasis? ब्लड से तो रिलेटेड है, बट देयर आर 1000 थिंग्स रिलेटेड टू ब्लड. Yes, very good. कहाँ पढ़ा? फिजियोलॉजी में. इसको और डिटेल में भी पढ़ाएंगे. And there, then in hematological practicals ना, they'll teach you there. There are certain disorders related to that also. BTCT, bleeding time, clotting time. So this comes with a just brief overview. I am saying a brief overview, okay? Each and everything would be covered again separately in detailed classes, in biochemistry also, in physiology also. They'll also take. Just one final food for thought for each and every one of you. In facilitated diffusion, each and everyone says that it does not require energy. Still the transport is going on involving a carrier molecule, which does not require any energy. But where does it gets the energy to transport against the concentration gradient? How? मतलब concentration gradient है. There is a carrier protein also there. the particle must move against along the concentration gradient as in diffusion. There is nothing involved. But where that carrier molecule is deriving that energy to change its conformation, पहले ऐसे है फिर ऐसे, passing through that, वो कहाँ से आ रही है? सोचा कभी किसी ने? Obviously, it's a carrier protein. It's require change in shape, and changing shapes of organic molecules require a very tedious process. Anyone thought of that? किसी ने सोचा कभी? That is also derived from the free energy, the Gibbs free energy present in the system. That energy of the change in conformation of the carrier molecule is derived from the free energy available in the system. It's a bit complex term, but you should know this. थोड़ा सा ये बात complex है. अगर मैं इसे दोबारा explain करूँ ना, तो you can see कि in diffusion particles just move from higher to lower concentration. ठीक है? High से low में particle तो चले गए since it has a concentration gradient. It in facilitated transport, it is a carrier molecule which is flipping its side. ठीक है? Now, from where this flipping is getting, from where this carrier protein is deriving its energy for flipping, it is deriving through the Gibbs energy present in the system. Okay? So, याद रखिएगा इस बात को. हालांकि इट्स बहुत ऊपर की बात है, मतलब शायद आप लोगों के इस लेवल पे तो फर्स्ट ईयर में तो काम ना आए, बट ऑलवेज मेक अ हैबिट ऑफ राइटिंग दीज स्मॉल पॉइंट्स. ओके, these help a lot. So this comes with the end of the lecture. Any queries, any questions, any doubts, you are free to ask. Stopping of blood flow. कट लगता, इट्स अ काइंड ऑफ, यू कैन से कितने टाइम में प्लेटलेट प्लग फॉर्मेशन हो जाती है. द फर्स्ट स्टेप इज फॉर्मेशन ऑफ प्लेटलेट प्लग. ओके, फॉर्मेशन ऑफ प्लेटलेट प्लग. वंस दैट प्लग फॉर्म्स, bleeding stops, and that is called hemostasis, not homeostasis.